Just 8 miles to the east of Dusseldorf, a city renowned for its fashion, World Fair and fine art, and who could possibly forget Germany's premier electronic band Kraftwerk, is a rather unremarkable little kiln that marks the site of a small cave where limestone was quarried. Quarrying was active back in the mid-1850s, but the little cave was lost till it was rediscovered after a determined search back in 1997. The reason why such a search was mounted was the discovery back then of fossils that were uncovered by 2 Italian miners in August of 1856. The cave was about 20 metres above the valley floor. The Kleine Feldhofer Grotte, as it is now known, had a 1 metre opening and was about 3 metres wide by 5 metres long by 5 metres high. From it was unearthed a skull cap and several bones. I say unearthed and not discovered because these precious finds were dumped with debris from the cave down the side of the valley. Fortunately, the fossils came to the attention of the owner of the cave, one Wilhelm Beckershoff, who, thinking they belonged to a prehistoric bear, took them to a local school teacher, John Carl Furlot, who was also an avid fossil collector and naturalist. In total, around 16 bone fragments, including the impressive skull cap, were recovered from the little cave. Furlot, recognising the bones, came from something a lot more human than a bear. A report of the find was published in the local newspaper dated September 4th, 1856, that would attract the attention of 2 anatomy professors at Bonn, Professors Hermann Schaffhausen and August Franz Joseph Karl Mayer. They recognised the antiquity of the find and called attention to the strong brow ridges above the eyes and the low sloping forehead, reminiscent of the great apes. They concluded that it belonged to a native tribe that lived in Germany before modern humans in antediluvian pre-biblical flood times. It would be not till 1864, though, that the specimen would be given the name we are familiar with today, by Irish geologist William King. King would later decide that they didn't deserve to be included in our own genus, though. The fossil found in the Neander Valley is the type specimen for Homo neanderthalensis, Neanderthal 1. A type specimen acts as a reference when species are first described. In truth, though, Neanderthal 1 was not the first specimen to be found. That honour goes to Engis in Belgium. Here, in 1829, fragmentary remains of a 2 to 3 year old child were found by Dutch naturalist Philippe Charles Schmerling. At the time of discovery, though, even though considered to be old, they were regarded as being the remains of ancient Homo sapiens. But just who were these Neanderthals? A species in their own right, or just a thick-skulled variety of Homo sapiens? In this episode, let's have a closer look and ask, would you recognise Neanderthals at the supermarket? Were Neanderthals brutish cavemen? Where did Neanderthals fit in our family tree? And what happened to Homo neanderthalensis? One of the first reconstructions of a Neanderthal came from a relatively complete skeleton that was discovered in 1908 at the La Chapelle-aux-Saints cave in central France. A specimen, a male, is regarded to have died about 60,000 years ago at the relatively ancient Neanderthal age of about 40 years old. The old man of La Chapelle was studied by French paleontologist Marcelin Boule, and it is from him that we gain our first reconstruction of the species. Boule commissioned a reconstruction that portrayed Neanderthals as brutish and ape-like. He even provided them with a slouching gait, drawing sharp contrast with proud, upright walking Homo sapiens. The image was printed in the Illustrated London News, who praised the artist, Mr. Kupka, for having given the face the expression it must have worn. As you can guess, it had the face of a wild animal and not a wise man. It has been suggested that the condition of the old man when he died may have confused Boule. At his death, he was suffering from osteoarthritis, which may have led to an interpretation of that slouching gorilla-like gait. But it is likely that what we are looking at here is more a case of cultural bias than misinterpretation. Interpretation being guided by the preconceptions of an ape man. This brutish vision of Neanderthals would persist for many years. The species was regarded as extremely primitive when compared to Homo sapiens, and not very closely related to us at all. It would appear that many of the scientific community at the time would have liked to have kept this part of our family strictly at arm's length. So, what is our current view of Neanderthals? If you were to meet one in a supermarket wearing modern clothes and with a tidy haircut, would you pay them a second glance? If you were to meet a Neanderthal, possibly one of the first things you would recognise is their heavy-set face. They had pronounced brow ridges below a sloping forehead, a rather receding chin and a large prominent nose. They had large eye sockets with areas of the brain devoted to processing vision. This has led some to suggest that Neanderthals had particularly acute eyesight. Neanderthals were on average a short species, with males around about 164 to 168 centimetres and females about 152 to 156 centimetres tall. At 166 centimetres, I may have been able to play for a Neanderthal basketball team, but meeting them in a football field or a wrestling mat would have been a little scary. Although they were short, they were packed with muscles, perhaps built a bit like Olympic powerlifters. They might have looked a little stubby in the leg, having femurs proportionally shorter than we have, but they possessed particularly strong arms and hands. They probably hunted large prey at close quarters using spears that they would lunge with rather than throw from a distance. Genetic studies, more on this later, have shown that Neanderthals probably had light-coloured skin. Some may have even had red hair. The first true Neanderthals were thought to have evolved in Eurasia, a significant distance from the human cradle in Africa. In the north there was a selective advantage to evolve lighter skins with less melanin, which would allow for the synthesis of vitamin D with a less intense sun. Neanderthals were living in a much cooler world during the last ice age. This may in part explain their stocky build and adaptation to retain heat. It's also been suggested that the large schnoz of a Neanderthal was an adaptation to warming cold air before it entered the lungs, although some have questioned this, as a large nose would have also been a large area for heat loss too. So our overall picture of Neanderthals at the moment is a rather short, stocky, barrel-chested and powerfully built people. They had heavier faces than Homo sapiens, but were a far cry from the brutal, shambling ape painted by Boole. So even if Neanderthals didn't look like Boole's brutish ape, did they behave like them? Were Neanderthals just human-looking, or did they actually demonstrate human intelligence and complex social structures? If you consider pure brain size, Neanderthals actually have us beat. The average cranial capacity of Homo sapiens is 1,400 cubic centimetres, and Homo neanderthalensis is around 1,600 cubic centimetres. Brain size, though, is no guarantee of complex social behaviour. As we said, it is possible that a larger part of the Neanderthal brain may have been devoted to vision. So do we have any other evidence of intelligence? Perhaps we could take something from the Neanderthal diet. It would appear that they ate a variety of foodstuffs, including plants and animals, depending on season and location. As Neanderthals were not a tropical species, they could not rely on stable plant diets year-round, forcing them to rely more on meat than their tropical cousins. Neanderthal wooden spears have been found from sites where big game animals have been butchered. To hunt such large animals implies cooperation, social structure, planning, and perhaps the hallmark of an intelligent animal, some sort of complex language. Obviously, this is difficult to prove. Neanderthal DNA may help us here. We know that on chromosome 7 in the human genome, there is a gene called FOXP2. This is required for the development of speech and language, controlling the development of various features in the brain, heart, lungs and gut. Mutations in this gene can lead to speech and language disorders. There are only 2 differences in the amino acid code for the FOXP2 gene between humans and chimps, and between Neanderthals and humans, there is no difference. Does this suggest that they had inherited the gene from a common ancestor even further back in time? If this is the case, could we also infer that Neanderthals were equipped with the language production capacity that we humans have? Neanderthals were adept at making sharp flakes of stone for a wide variety of purposes, including cutting and scraping. Neanderthal tools have often been thought of as primitive when compared to those produced by Homo sapiens. However, they are still very versatile and efficient. At a former Neanderthal hunting campsite in eastern Germany, where butchered remains of various animals were found, something intriguing was discovered. It would appear that the Neanderthals had developed technology to attach points to spears using an adhesive pitch that they made from birch bark. In order to do this, they would have to employ a technique known as dry distillation to make a sticky, resinous goo, a process that requires temperatures carefully controlled around 343 degrees centigrade with no oxygen. This is not a simple process and would likely have required experimentation and an ability to imagine possible outcomes to create what was to be considered, at 40,000 years before the present day, one of the world's first industrial processes. But there are examples of social interaction that might also suggest an intelligent species too. In Shanidar Cave, located in the Brados Mountains in Iraqi Kurdistan, the remains of 10 Neanderthals have been found. A cast of one of those, Shanidar I, is held at the Smithsonian's National Museum of Natural History. He was an old man between 40 and 50 years of age who was certainly showing signs of his age. He was probably blind in one eye from a blow to the head. He also had a withered right arm that was fractured in a number of places. These fractures had healed, but he probably had very limited or no use of his lower arm and hand. He also walked with a noticeable limp. As these had healed, though, this implies care by his social group. This person, especially in his later years, would have probably required a lot of support from the members of his tribe. He was not abandoned, but thought of as important to the rest of the community and worth the effort of helping him live a relatively long life, even with his disabilities. The same was probably true for the old man of La Chapelle-aux-Saints. The individual here had osteoarthritis, had few teeth, and would have required help from others. When he died, this Neanderthal may have even been carefully placed in a shallow grave. This is not an isolated case and shows that thought was being given to the dead, a certain reverence and perhaps the concept of an afterlife, revealing the possibility of an ability to conceptualize. For a long time, one of the features of Homo sapiens, regarded as being fairly unique and distinct, was the production of art. Like the concept of an afterlife, art illustrates an ability to conceptualize, to represent the world we see or perhaps we don't see that is locked in our imagination. The Lascaux and Chauvet cave paintings in France are beautiful examples of this, the artists laying down fantastic images at 17,300 and around 31,000 years ago, respectively. The same goes for the small statuettes and figurines that are associated with the spread of Homo sapiens into Europe, such as the Venus of Willendorf. So, do Neanderthals exhibit similar artistic abilities? For a long time, it was thought the answer was no, but a geometric crosshatch pattern found at the back of a cave in Gibraltar in 2012 may change that. These symbols are thought to be around 39,000 years old and were discovered below an undisturbed layer of sediment in which Neanderthal tools were found. The image appears to have been made by a point, the artist deliberately deepening the cuts in the hard dolomite rock over many hours. This was not an accidental artifact of cutting meat or some other substance. And what does it mean? There are still some who question if these markings were actually made by Neanderthals, but if they were, this is significant. Perhaps the image is a map or perhaps a symbol to mark territory. Whatever its meaning, it shows evidence of abstract thought, and if this is the case, and these are produced by Neanderthals, then they are not unlike those produced by modern Homo sapiens. Other interesting evidence of the possible cognitive abilities of Neanderthals has come from a 2012 paper by researchers from Spain and Gibraltar. They studied bird bones, including jackdaws, vultures and eagles, found at over 1,699 Neanderthal sites across Eurasia. What they discovered were cut marks on the wing bones and evidence of peeling of the bird's feathers. Wings now are not nutritionally useful compared to the rest of the bird, suggesting that the dark feathers of these birds may have been collected, perhaps worn in some sort of decoration. If this is the case, this is once again evidence of complex thought. And add to this the suggestion that Neanderthals may have been using red and black pigments, perhaps to paint their bodies, and using shells as ornaments, and a much more colourful and thinking human emerges. So just how similar to Homo sapiens were Homo neanderthalensis? As we have seen, they looked different, but not as brutally different as originally thought in Boole's days. They appeared to care for the members of their kind and likely had significant communication skills and perhaps an appreciation of conceptual art. But what about at the deeper genetic level? Just how closely are we related to Neanderthals? Unlike reading DNA that is many millions of years old, like we would need to for the dinosaurs, the prospects of reading DNA that is within the hundreds of thousands of years range is much better. Perhaps we can read the genome, therefore, the genetic recipe of Neanderthals. It is important to realise that there are two possible sources of DNA that we can use in genetic studies. Firstly, nuclear DNA. That is the DNA found in the nucleus of our cells that codes for all the structures and functions of our bodies. This is where we store our blueprints, as it were. But there is another source of DNA in small micromachines or organelles that are found in our cells. These are mitochondria, and each of your cells contains around 1,700 of them. Mitochondria are essentially the power engines of eukaryotic life, that is, life more complex than bacteria or prokaryotes. It is thought that they originally were independently living prokaryotes that became engulfed but not digested by its host. The mitochondria got a home safe from the external environment, and the host cell got free power from the mitochondria. As a result, mitochondria have their own DNA, independent of the personal blueprint that you have in the nuclei of your cells. We call this DNA mitochondrial DNA. Sperm and egg cells also contain mitochondria, but when the nucleus of the sperm fuses with that of the egg to give you the mix of mom and pop genes that we're all composed of, the mitochondria of the sperm most of the time gets left outside. As a result, it is generally only the maternal mitochondrial DNA, that of the egg, that gets passed on down through the generations. Over time, mitochondrial DNA, like nuclear DNA, will undergo random mutations, slight errors when the DNA is copied from one generation to another. We can use these differences between mitochondrial DNA in different people and estimate the rate of mutation to determine how far back in time they once shared a common ancestor. We can also find out how long since they shared that ancestor that they've been travelling along their own particular branch of Ancestry Road. It was using this principle that researchers in the 1980s were able to determine that the common ancestor of all mitochondrial DNA, and therefore all humans living today, was a woman who lived in Africa around 100,000 to 200,000 years ago, mitochondrial Eve. Tracing the last common ancestor of humans and Neanderthals would be attempted by Svante Pääbo of the Max Planck Institute in Germany. For his study, they would use an old friend, Neanderthal 1, the type specimen of the species. Even if DNA is present, there were problems though. Most of the DNA would be fragmentary, and the vast majority of the DNA you would find would be from contaminants from soil bacteria, plus healthy doses from scientists and curators who have studied the specimen over the years. Taking these contaminants into account though, Svante's team finally isolated Neanderthal mitochondrial DNA and then compared it to around 1,000 mitochondrial DNA sequences of modern humans. Among the moderns, the DNA differed by about 8 mutations, but between the moderns and Neanderthals, the average was around 26. This would place the common ancestor of Neanderthals and modern humans around 500,000 years ago. Perhaps this guy, Homo heidelbergensis, who was known to have lived in Europe and Africa at about the time, is close to the split of the Neanderthal and modern human lineages. Some research, based on tooth structure, has suggested that the split occurred even further back in history, perhaps as much as 1 million years ago. This places it in the range of ancestral humans like Homo erectus. This ancient divergence was taken by some to suggest that Neanderthals were a completely separate species to humans, and that with around 500,000 years or more of genetic drift between the two populations, it would be very unlikely that a human and a Neanderthal could breed to produce living offspring, let alone offspring that might be viable enough to pass on genetic mixing into the next generation. There had been intriguing fossil finds, though, that suggested some interbreeding had occurred. For example, the Lapido child found in the limestone canyon of Lagar Velho in central Portugal. This skeleton of a human child was found in 1998, buried with red ochre and pierced shells dating to about 24,500 years before the present day. Although looking like a modern human, the skeleton contained some rather Neanderthal features. Was this a hybrid, evidence of gene mixing between sapiens and Neanderthalensis? Or was it just a stocky child? Had there been interbreeding despite the antiquity of the last common ancestor of the two species? To answer this, we would have to find some way to read the nuclear DNA of Neanderthals, the blueprint of the organism held in the cell's nucleus. This was the quest for the Neanderthal Genome Project. This is something which we have only just recently achieved for our own human species in the Human Genome Project, an international effort to read our own human blueprint. But just what is a genome? Well, basically, a genome is all the genes that themselves are composed of DNA, the famous replicating molecule, that code for any organism. The code itself is written in an alphabet of just 4 letters made up of 4 molecules, 8 to 10 atoms wide. These nuclear bases are adenine, thymine, cytosine and guanine. The nuclear bases exist as base pairs with A bonding with T and C bonding with G. The order in which these letters appear on the DNA strand code for specific amino acids and thereby proteins in the cell. The most common way to determine the sequence of these letters is to use an enzyme called DNA polymerase, an enzyme that synthesizes DNA in our cells. To sequence a piece of DNA, the enzyme is used to generate a new strand of DNA, but using nucleotides that have been tagged fluorescently. This fluorescent signal can be read, the signal varying depending on which nucleotides were used. Short, overlapping segments are read, and from these are longer sequences constructed. The Human Genome Project was initiated in 1990 and completed in April of 2003, an incredible step forward in understanding who we are at our very core. It will hopefully provide new insights into diseases like cancer that have plagued us for so many years. But what of the Neanderthal genome? This would be a more challenging endeavour. There would be all the problems with contamination and degradation of the DNA that we mentioned earlier. There would also be the added issue of distinguishing Neanderthal DNA from a very close relative, us. These were the challenges facing Svante Pääbo's team at the Max Planck Institute when the Neanderthal Genome Project was initiated in July of 2006. Some of the first specimens to be studied came from the femurs of 3 female Neanderthals found in the Vindija cave in northern Croatia, dated to about 38,000 years old. The bones were well preserved, suggesting that DNA might be recovered. Using enzymes that eliminated bacterial DNA, the team was able to concentrate the Neanderthal DNA by about 5 times. Eventually, a Neanderthal genome was sequenced with an initial draft published in the journal Science in 2010. This now allowed for comparisons to be made between humans and Neanderthals at the level of their DNA. It was concluded that around 98.7% of the base pairs in the 2 genomes were identical. It was from this that comparisons between the FOXP2 gene in humans and Neanderthals could be made that we mentioned earlier. but something else too. In comparing the Neanderthal genome to people from different racial groupings, they found around 1 to 4% of Neanderthal DNA is in our genome. The only way it could have got there is via interbreeding. Despite the distance in time that separates humans from Neanderthals from their last common ancestor, it would appear we were still sufficiently similar to produce viable offspring. There is more to be gleaned from the data though. No Neanderthal DNA has been found in Africans, suggesting that as modern humans migrated out of Africa, they met and occasionally bred with Neanderthals that they came across. Not everyone has the same part of the genome, but in total it is thought that around 30 to 40% of the Neanderthal genome is floating around in the human population. This indicates that there was not only one Neanderthal ancestor, but there must have been an entire history of Neanderthal-human interactions. As I said, any non-African today has between 1 to 4% of Neanderthal DNA, but a discovery by researchers including Svante Pääbo from the Max Planck Institute, reported in the journal Nature in 2015, provides data from an individual much closer to the original interaction. A specimen from a cave in Romania, dated to 37,000 to 42,000 years before present, has been found with between 6 to 9% Neanderthal DNA, probably indicating a fully Neanderthal ancestor just 6 generations back. Today, Neanderthal DNA is found to be more common in certain places in our genome than others. For example, around 80% of Eurasians have a Neanderthal keratin gene, possibly advantageous to those members of our African-originating species who are moving into colder environments. In addition to skin toughening, we may have also received a boost to our immune systems, resistance to certain diseases that Neanderthals had battled for generations before they met modern humans. It's not just good things we get from this interbreeding though. It's possible that certain undesirable traits have been introduced into the mix, including lupus, Crohn's disease, and an increased chance of developing type 2 diabetes. In the case of diabetes, this could be the result of what was originally a useful trait. Neanderthals were hunter-gatherers living through times of plenty, but also scarcity. There would be a definite advantage for individuals to have an ability to gorge themselves when food was available, to make up for time when food was scarce. Useful back then, but dangerous today in the West, where food is plentiful and available 24/7. It is important to remember, though, that the overall negative impact of our Neanderthal DNA is probably very, very small indeed. This then is one possible picture of our messy genome that was published by Chris Stringer of the Natural History Museum in London in the journal Nature in 2012. Humans and Neanderthals diverged from a common Heidelbergensis ancestor with Neanderthals, generating another ancient human group, the Denisovans. Each of these forms included an archaic flow back into the Homo sapiens population, contributing in varying degrees some small parts of the human modern genome we see today. So what happened to the Neanderthals? The forerunners of the species arrived in Europe about 800,000 years before present. Homo sapiens arrives at about 40,000 years before present, but just 10,000 years later, the Neanderthals are gone. This could suggest that they were outcompeted for resources, or, a darker view, were hunted down and exterminated in acts of interspecies genocide. Another possible explanation may relate to rapid climate change. By about 50,000 years ago, climate started to fluctuate rapidly from extremely cold to mild within a few decades, perhaps within the lifetime of an individual. This would have caused rapid changes in the landscape from woodlands to grasslands, with familiar plants and animals appearing and disappearing. It's been suggested that early humans during this period would have had more widespread social networks. This would have allowed them to acquire resources over a greater area. Neanderthals in this model become increasingly isolated and starved of resources. They finally become extinct at about 41,000 to 39,000 years before the present, at the start of a very cold snap. But perhaps there is another explanation. As modern humans moved into Neanderthals' territory, perhaps the Neanderthals were just absorbed by interbreeding. Or perhaps there was a combination of all three, part confrontation, part climate and resource related, and part genetic assimilation. For myself, I'm happy to think that at least genetically, a part of these ancient peoples are still with us today.